session, I request Professor Kiyoshi Yasukawa for the session of invited talks. Sir is professor at Graduate School of Agriculture, Kyoto University, Japan, in the Laboratory of Enzyme Chemistry. Sir has numerous publications in various international peer-reviewed journals like J of Biochemistry, Protein Engineering Design and Biosciences, Biotechnology, and Biochemistry. Sir is going to deliver his talk on the topic genome profiling based mutation assay using a mammalian cell. So please join your hand together and welcome Sir for his talk.
Please show this slide. Good morning, everyone. I'm very happy to have an opportunity to make a presentation here in Biyani's college. Actually, it's my first time to come to India. First of all, I'd like to briefly introduce Kyoto University and my division with a few slides. This is the entrance gate, main entrance gate of Kyoto University. Kyoto University has a lot of faculties and graduate schools. Dr. Nomura, the previous speaker, belonged to Grad School of Education. In my case, I belong to Graduate School of Agriculture. Graduate School of Agriculture has 7 divisions, and I belong to Food Science and Biotechnology. In this division, mission to establish fundamental concept of foods and improve quality of life from various points of view. We are taking various approach. And this division has 8 laboratories, and I'm in charge of enzyme chemistry. As you know, the word enzyme comes from Greek word enzyme, in yeast. Enzyme are proteins having catalytic activity, as all of you know. It accelerates and accurately controls biological reactions in organisms. And enzymes are widely used in industry. When bound to substrate, enzyme converts the structure of substrate into product. Actually, I have done a lot of works in this field, but today I'd like to concentrate on GPMA method. This is the title of my talk today. GPMA, Genome Profiling Based Mutation Assay. Very complicated, using a mammalian cell. GPMA is a method which detects a mutation of genomic DNA caused by mutagenic chemicals without sequencing and with high sensitivity. The minimum detection level is as low as 10 ppb. The reason why I have chosen this topic is that this technique was developed not by me, not by I, but by these 4 persons. Dr. Koichi Nishigaki, a professor of Saitama University, and Dr. Miho Suzuki, she will make a presentation today after I, now associate professor of Saitama University. And as you know, Dr. Manish Biyani used to belong to Dr. Nishigaki's laboratory. And Parmila Kumari, do you remember her? Today, Dr. Manish Biyani introduced her as one of the successful students who graduated from Biyani's college. So after here, she joined Nishigaki's laboratory and involved in GP method and got a doctorate degree. Now he works as a postdoctoral fellow in Japan. And I did this work this year, and she was our teacher actually. So we learned that all techniques from Dr. Kumari, Parmila-san, and the advisor of Dr. Nishigaki. GPMA consists of 4 steps. This slide shows the first 3 steps. The first is culturing. E. coli is cultured with or without a chemical for 15 generations. Then genomic DNA is prepared. The second step is random PCR. Please raise your hands if you have done PCR. Almost all people have never done PCR. So it's a very one of popular technique to amplify target nucleic acid sequence. And in this method, random PCR is applied to amplify some fragment from genomic DNA. Amplified DNA is applied to zero electrophoresis. Please raise your hand if you have performed zero electrophoresis. Okay. In this zero electrophoresis, temperature gradient is applied. So DNA has various forms depending on temperature. Low temperature, perfectly double strand. But with increasing temperature, such a kind of melting, and finally, it's completely separated into single-strand DNA. So amplified product gives such a kind of profile. The last step is processing. We analyze the profile. E. coli, before the culture, exhibit this profile. E. coli, after the culture without a chemical, exhibit this profile, which is similar to that. In the case of E. coli, after the culture with a mutagenic chemical. it shows profile different from that, because the DNA has a lot of mutations. And it's very difficult to be explained. So such a kind of difference can be quantitated due to these equations. Finally, delta paths reflect the mutagenicity. This system was established by Dr. Nishigaki's group with Dr. Suzuki and Dr. Biyani and Dr. Kumari, Parmira-san. And this shows a comparison of Ames test and GPMA. Ames test is, as far as I know, in Japan, a golden standard of mutagen assays. If this substance is mutagenic, that means very dangerous or not. It's determined by Ames test in Japan. It's a governmental approved method. Ames test detects a specific point mutation within the histidine gene of Salmonella after the culture with a chemical substance to be tested. Because this test targets only one gene and at a specific site, so the detection sensitivity is low. The minimal detection level is 1 ppm. In contrast, GPMA detects all the point mutations with random PCR amplified DNA. So it has better sensitivity. The detection level is 1/100th. This is the structure of AF2, one of famous mutagenic chemicals. In GPMA, so the difference mutagenicity can be detected at 10 ppb by the difference delta path. In Ames test, such a kind of difference cannot be detected. But DPMA, the results well correlate with those of AM test, except for these chemicals. Both GPMA and AM test had good correlationship. The purpose of our project this year is, in the conventional GPMA, Escherichia coli has only been used, cultured with chemical substances to be tested. And the mutations incorporated in the genomic DNA are identified by the change in the pattern of gel electrophoresis. However, mammalian cell is preferable to E. coli, considering the use of the result for the estimation of mutagenicity to human, because chemical substance receives enzymatic reaction inside cells. So in this study, we used mouse NIH3T3 cells in GPMA instead of E. coli. Material method I'd like to skip. Culturing followed by preparation of genomic DNA and PCR and gel electrophoresis. We use this cassette for electrophoresis. And this is a typical pattern of the profile of electrophoresis. This slide shows the effect of mutagenic ethylium bromide. I think some of them have used these chemicals to see nucleic acid. But it's very dangerous on the growth and mutability of NH3 cells. As you can see this panel, this photograph, without these chemicals, the cell grew very well. But with 10 ppm, the cell didn't grow at all. So very mutagenic, very dangerous. In GPMA, even at a low concentration of 10 ppm, without ethylium bromide, the cell grow well. But with ethylium bromide, it didn't grow well or it didn't grow at all. And in GPMA method, delta path increased with increasing chemicals concentration. We did additional experiment using these 5 chemicals. The number represents the name of reagent, chemicals. And in Ames test, these 4 chemicals are positive, mutagenic, while the rest of 2 chemicals are negative. The blue bar indicates the result of GPMA with NYH3T3, and gray bar, the result with E. coli. Delta path obtained in GPMA with NYH3T3 well correlated with that obtained in GPMA with E. coli, except for number 3. The discrepancy observed in number 3 might be due to the difference in intracellular metabolic network between these 2 organisms. We also examined the correlationship between 2 GPMAs using 20 chemicals. Open triangle means Ames test mutagenic chemicals. Filled one represents Ames test non-mutagenic chemicals. As you can easily see, delta path obtained in GPMA with cells correlated with the obtained in GPMA with E. coli. The regression coefficient is as high as 0.77. So all results suggest that mammalian NYH3T3 cell can be used in GPMA. This is a summary. So the reason why I chose this as a topic of today's presentation is that these techniques was developed by Dr. Nishigaki's group, including Biyani sensei and Parmila-san, the graduates from Biyani's college. But another reason is that I now have a future research plan using this technique with, in the collaboration with Dr. Biyani and Dr. Teres, the last speaker. Okay? Okay, thank you for attention.
Thank you very much, Yasukawa Sensei, for making the introductory part of GPMA. I think many of you have realized that this technology is developed by one of you, Pamela Kumari, who was just like you in 2009. And then she went and she developed this technology. And this technology now can use to detect the genomic variation in our body. And then can be, I think it's equivalent to sequencing. Those who are familiar with DNA sequencing, this technology is equivalent to DNA sequencing and can give the all information of genetic structure in your body, in your single cell. So I think this is a revolution in this technology, I think, to have this GPMA. So now we are open for 1 or 2 queries from students. If you have anyone? Okay. Can you hand over the mic?
Good morning, sir. Sir, why NIH 3T3 cell is used?
Why? The reason?
Yes.
Sir,
The reason? I think any cell can be used. The reason why we use this cell is that this is a mouse fibroblast cell, and very famous and very easy to be obtained. So that's why we chose this cell. So in other words, we don't have a specific reasons. But now we are now doing the same experiment using liver cells. I think in some cases, the difference in cells lead to the difference in mutagenicity due to the difference in metabolic system in cells.
thank you, sir.